Hi there, my name is Lindsay Roberts, and I will be presenting on the anatomy and enzymology of fat digestion. First of all, I want to start with a discussion of the basic anatomical components of fat digestion and the different parts of the digestive system that are involved in fat digestion. So I included a diagram just to show the basic components, but just wanted to give a quick description of what each of them accomplish. So when we look at fat digestion, a huge component would be the bile secreted by the liver cells. Now, when the liver secretes this bile, it travels to the gallbladder, and what stimulates this release would be contraction of that gallbladder during digestion. This is the stimuli that kind of triggers the bile to be sent to the small intestine via the common bile duct. Now, this isn't just a constant transmission of bile. There is a component called the sphincter of Oddi that moderates this release of bile to the small intestine. Once that bile is sent to the small intestine, its purpose is to digest lipid, and it eventually travels to the ileum of that small intestine. Really, the production of bile and its digestion is more of a cycle. So once the bile digests these lipids, it's actually recycled back to the liver, and that's via the hepatic portal vein. After this occurs, the bile is actually reabsorbed by the liver cells to be reused during the next digestive cycle. Lastly, just wanted to mention this diagram talks about the hepatic artery, which essentially brings oxygen to the tissue so that it can carry out all of these functions. Now, in terms of the more mechanical components of digestion, the food that is traveling to the intestine includes lipids that don't really have enough surface area to interact with the enzymes that basically allow for digestion. And this is because of their structure, their chemical structure. Most often, digestible lipids are what are called triglycerides, so fat from animals. So to be able to be digested, these molecules need to be broken down into more stable molecules, and this essentially increases their surface area so that they can bind to enzymes, and also to bile. So I included a diagram of the kind of what triglycerides are made up of. So essentially they have that glycerol backbone and then have 3 fatty acids bonded to them. So when this molecule is broken down, it's going to break down into glycerol and then free fatty acids. Now, when it comes to bile salts are comprised of a polar portion and a nonpolar portion, which allows bile to essentially connect with those lipid molecules. Now, when it comes to the role of the enzyme, an enzyme called lipase is what breaks down that triglyceride into a monoglyceride and 2 or 3 fatty acids. Kind of what I talked about in the last slide. Now, once lipase breaks down that molecule, it has kind of met its purpose. And then another enzyme comes into play to bind to the actual lipid molecule. So that enzyme is called colipase. Now, what that enzyme does is actually displace the bile salts so that that lipid portion of the molecule can be reached. And essentially, what occurs is that this micelle is created, and it's kind of this bundle of enzymes and bile salts, the fatty acids that have been broken down, the monoglyceride, and cholesterol. It's all bundled into this micelle. Now, when it comes to the absorption of these broken down molecules, this micelle travels to the brush border, and eventually the lipids break off from that micelle and enter the cells via simple diffusion. So it's almost like the micelle is just carrying the lipids to be absorbed. Now, once these lipids have entered the enterocytes, they basically travel into the cells to the endoplasmic reticulum, and there they become triglycerides again. And essentially these triglycerides then go through a second binding process, and they combine with cholesterol and proteins to form what are called chylomicrons. Now, this new bundle of materials, of molecules, travels to the Golgi apparatus in a vesicle to basically then leave the cell now that it's a chylomicron, and this occurs via exocytosis. Once this exocytosis has occurred, the chylomicrons have left the cell, and they are reabsorbed by the lymphatic system. So these lymph vessels called lacteals pick up those chylomicrons, and they transport those chylomicrons basically through the lymphatic system and into the venous supply, and basically those digested fats reach the heart, which in large amounts can result in some serious health issues. Now, that concludes my review of fat digestion. Thank you for listening.